Hello, my name is Guadalupe Bolaños, and my presentation will be over fluorescence resonance energy transfer. So just a little bit about it. It's called Foister or fluorescence resonance energy transfer, and in this presentation I'll just refer it as FRET. And it studies the interactions between integral membrane proteins and cellular membranes. And it's mostly applied in single molecule experiments, but it's also applied in molecular motors, biosensors, and DNA mechanical movements. So basically, energy is transferred from an excited donor to an acceptor group through a non-radiative process, and your donor and acceptors are referred as a fluorophore, and they come from a class of autofluorescent proteins called GFPs or green fluorescent proteins. So the general application is to have energy transferred non-radiatively from an excited fluorophore to another fluorophore referred as the acceptor by means of intermolecular long-range dipole-dipole coupling. And they usually measure the distance between two positions of interest on a big molecule by attaching appropriate donor acceptors to that molecule. And there are two different approaches to FRET that I read, and one was emission measurement, where you record the excitation of your donor and you detect the light emitted by either the donor or your receptor in the presence of another fluorophore. So basically the donor emission you basically see the donor emission being decreased and the acceptor emission being increased. Or the other approach is acceptor photobleaching, which records the excitation of the donor and detects the light emitted before and after the acceptor photobleaching. But both of them provide a good FRET index or a good transfer efficiency. So I found this figure in an article that wanted to see if we could use FRET not just only in vitro but also in vivo. So the key question here of this figure was to specifically look into validating the biosensor of FRET in cultured cells in order to make sure it would monitor the activity of the target kinase. And so the experimental design, they had a positive control. They used HeLa cells expressing protein kinase A biosensor, and a negative control group that had serine and threonine kinase FRET biosensor, which was a mutant in which photoacceptor, the serine and the threonine were replaced with alanine, and you see here the name. And both of these were stimulated with one of a dibutyryl cyclic AMP. And what they found was that the FRET/cyan fluorescent protein ratio gradually increased upon the addition of the stimulant, indicating pKa activity. And so pKa activity is stimulated by cyclic AMP, and it just basically makes it switch on signal transduction pathways or to either phosphorylate proteins. And you can see that as more time passed, the cells got more stimulated. And on this one, which was which had replaced its amino acids, no stimulation occurred, so it showed no increase. It just acted the way, both of these acted the way they were supposed to. So some limitations of this study was that you have to carefully evaluate and characterize the FRET biosensors before use, and you cannot select cells that express extremely high or low levels of biosensors. And you have to be careful when photobleaching because it can lead to artificial decrease of FRET imaging. But some of the strengths were that it was able to show the quantitative relationship between FRET increase or decrease and the net change of intracellular signaling. And it's very good for spatial and temporal dynamics of intracellular signaling in cells. So the question that I have for you is, what are some of the biological applications that use FRET imaging? What are some of the current studies that are using FRET as their technique? Thank you, and I hope you learned a little bit about fluorescence resonance energy transfer.